Now, glucose, as you all know, glucose is the most common source of cellular energy and a substrate for many biological processes. Because every cell in our body, it needs glucose. Glucose is the universal fuel for all of our cells. So how these cells will get glucose inside? So the glucose, since it's an hydrophilic molecule, so it needs a transporter. And the transporter for glucose is glucose transporters. There are a variety of glucose transporters. We can say glucose transporter 1 to glucose transporter 14. So especially glucose transporter 1, glucose transporter 4 and all that. So glucose basically gets into the cells through a glucose transporter which facilitate entry of glucose into the cell. So once it gets in there, so what happens? So glucose will undergo a metabolism where it breaks into different molecules, and in the process it gives energy. Okay. Now higher glucose utilization will be done by tumor cells, highly proliferative cells, infected cells. They all will take uptake of higher amount of glucose. That's why they will express increased number of glucose transporters over the surface. They will express increased enzymes, metabolic enzymes to degrade the glucose for energy purpose. Now, according to some estimates, there will be around like 20 to 30 fold increase in the uptake of glucose, increase in the uptake of means expression of enzymes in these cells, which are tumor cells, highly proliferative cells, infected cells like viral infection especially. Now, once inside the cell, so glucose enters into various cycles including glycolysis, and it will produce ATPs. Even it has been established in the cancer cells. It is a well established fact in the cancer cells. We call that as Warburg effect. So what it says is, even in the absence of oxygen, generally what happens in our cells, there will be oxygen present and glucose breaks down into pyruvate. Pyruvate goes into TCA cycle and generate ATPs, as you are seeing here in this particular slide. So that happens in the differentiated cells. In the presence of oxygen, glucose gives a lot of ATPs, like 30 ATPs per molecule, whereas even in the absence of oxygen, glucose being converted into lactate, it gives 2 ATPs. That's called anaerobic glycolysis. Now in the proliferative cells, what happens? Glucose, even in the absence of oxygen, whether it is present in the presence of oxygen or in the absence of oxygen, it will preferentially is diverted into lactate, and then lactate undergoes further metabolism, and it facilitate the energy needs of tumor cells or highly proliferative cells or infected cells, and also it will facilitate so many other metabolic changes, which we call it as cellular reprogramming, and thereby tumor cells will proliferate. I am going to take you into that aspect soon. Okay, now let's come to look at like what exactly is 2-deoxy D-glucose. As you see here in the figure, here is the glucose molecule. Let me highlight that glucose molecule. Here, this is the glucose molecule, D-glucose molecule, which is a universal fuel for our cell. Whereas as you see here, this is a D-glucose molecule. So because second carbon, especially the second carbon, do not have hydroxyl group here. Second carbon do not have hydroxyl group. Compare that with the glucose. This is the glucose here, second carbon having hydroxyl group. So deoxy, no oxygen atom there. So second carbon not having oxygen atom in the glucose, that's what is 2-deoxy glucose. That's the only difference between glucose, D-glucose, and 2-deoxy D-glucose. Okay, so glucose is a dextro sugar, that's why we often call it as D-glucose, D for dextro. So D-glucose and 2-deoxy D-glucose, difference is second carbon do not have hydroxyl group. That's what is 2-deoxy glucose, 2-DG. Now this is a synthetic analog of glucose molecule. What is the mechanism of this 2-deoxy glucose molecule now? Now the 2-deoxy glucose, since since it's similar to glucose molecule, which we call it as D-glucose analog. So what happens is it is also getting into initial reactions in glycolysis, where glucose is converted into glucose 6-phosphate by hexokinase enzyme. Similarly, by the same hexokinase, 2-deoxyglucose is also phosphorylated at 6th carbon and become 2-deoxy-D-glucose 6-phosphate. So glucose is converted to glucose 6-phosphate. Like that, 2-deoxyglucose is also converted into 2-deoxy-glucose 6-phosphate. This is done by hexokinase. Then what happens? 2-deoxyglucose 6-phosphate do not undergo further reaction in the glycolytic pathway usually done by phosphoglucoisomerase enzyme or glucose 6-phosphate isomerase enzyme. This enzyme do not convert 2-deoxyglucose 6-phosphate into fructose 6-phosphate. That's the job of enzyme. Like glucose 6-phosphate is converted to fructose 6-phosphate, whereas here 2-deoxyglucose 6-phosphate is not converted to fructose 6-phosphate, and that leads to accumulation of 2-deoxyglucose 6-phosphate. Then what happens? This 2-deoxyglucose 6-phosphate, it will have a inhibitory effect, feedback inhibitory effect on hexokinase enzyme, and also it doesn't go down into the glycolysis process. Thereby, it's going to inhibit glycolysis. Glycolysis is inhibited by having an inhibitory effect on hexokinase, thereby glycolysis and glucose metabolism is affected. Okay, and that's what is the mechanism of 2-deoxyglucose. Now, so the effect of this 2-deoxyglucose on glycolysis, it has been explored, like whether we can use it to treat different types of cancer. Okay, that's where it came from. That's why 2-deoxyglucose molecule was initially designed, and also this 2-deoxyglucose molecule has been used as a diagnostic tool. I'm going to come to that point soon. Okay, now 2-deoxyglucose is relatively non-toxic and orally available. It is an attractive tool for potential anti-cancer therapy. Its diagnostic potential has been widely explored, like in PET scanning, positive emission tomography scanning. Now let's look into what are the biological activity of 2-deoxyglucose in cancer cells. Okay, so 2-deoxyglucose, as I explained to you earlier, it is going to inhibit glycolysis. I have explained you the mechanism. And also 2-deoxyglucose, it causes autophagy induction. Autophagy is a degenerative mechanism present in every living cell. In general, autophagy is a process of sequestration of intracellular entities within double membraned vesicles called autophagolysosome and their delivery to lysosomes for degradation. In cancer cells, autophagy has a dual role in tumor development and survival. Autophagy enhances tumor growth in the early growth phase, and in advanced tumor increases cancer cell survival, and it promotes metastasis. Now, the third use of 2-deoxyglucose in cancer cells is it causes induction of apoptosis. Note that apoptosis is a programmed cell death. So, prolonged autophagy and extensive self-degradation ultimately leads to apoptotic cell death. It appears that potent 2-deoxyglucose apoptosis induction is transmitted via various pathways like autophagy, intrinsic and extrinsic pathway, and that depends on cellular type. Now, the fourth mechanism of biological effect of 2-deoxyglucose on cancer cells is inhibition of protein N-glycosylation process. You know, proteins, we have O-glycosylation and N-glycosylation. So, 2-deoxyglucose, it has got an inhibitory effect on N-glycosylation because its molecule is similar to mannose. So, 2-deoxyglucose, it kind of competes with the mannose in glycosylation process because N-glycosylation, we need mannose in sufficient amounts, and interference with the mannose with the 2-deoxyglucose will have a inhibitory effect on N-glycosylation, and further it leads to stress, oxidative stress and other it interferes with the biological effects. Okay. Now let's look into effect of 2-deoxyglucose on cancer cells. So, 2-deoxyglucose has the following effects on cancer cells. This is in summary. So, it inhibits glycolysis and ATP synthesis. It disrupts N-glycosylation process. It decreases energy metabolism and NADPH levels. It interferes with cellular thiol metabolism. It increases oxidative stress, and with all the above effects, 2-deoxyglucose appears to be an efficient cytotoxic agent. Importantly, all of these effects are mostly observed in cancer cells, and that too without significant effect on the viability of normal cells. That's what is the effect of 2-deoxyglucose on cancer cells.